Okay, for this second session, I'm quite excited by this, but because we have for the first time an industry session, and we were going to start up with Bonnie Brayton, and she's going to show us a little bit more a perspective from an investor point of view. So, good luck.
Thank you, Vincent. Okay, I'm going to start off by making a plug for Vincent. If you ever need a trivia night partner, Vincent is your guy. He knows more random facts than 80% of dads that I've met, which I learned last night. So high praise. Okay, here's my next joke. Prepare yourselves to laugh. I am bringing you, as your AgTech investor, I am going to bring you your lowest tech presentation here. There will be no slides. Brace yourselves. I'm just going to talk this whole time. We'll go on a journey together. So I'm going to start by introducing myself so that you understand the perspectives that I have on investing. Okay, I'm ready. Bonnie Brayton. I call myself a plant biochemist, but that's really inaccurate. I'm not an expert like many of you in the room. I'm more of a generalist in different areas of science. I started off as a chemistry undergrad. I did organic chem research. I did aerosol chemistry research in atmospheric contexts. When I went to grad school, I moved into plant biology, but specifically I studied plant-pathogen crosstalk. And in that molecular crosstalk between plants and pathogens, I wasn't actually doing pathogenesis or anything cool like that. I actually just did protein biochemistry. So that's my academic background. From there, I did many other random things. I, after grad school, went to a small biotech company of about 100 people. We did, our team in that biotech company did the biological chemistry for the company. Some of you might be aware of it. We have at least one other expat here today, this week. I worked for Mendel Biotechnology. Mendel had cloned all of the transcription factors in Arabidopsis, and then they made overexpressing lines for each one of those transcription factors. And what did they do with this? They worked with the multinational companies. So if a multinational company had a gene lead that they were interested in that was a transcription factor, they would ask Mendel to model what they did in Arabidopsis for their mode of action studies. So in that context, at my first startup, I worked with major companies, but from a startup perspective, and again, doing biological chemistry. From there, I took a personal pivot. I moved from the Bay Area to my husband's hometown of Honolulu, Hawaii. And there I couldn't get a job at Monsanto or Pioneer because I didn't drive a tractor and I couldn't speak Tagalog, and those were the two skills I needed to land a job at that time. So I went and I worked for the John A. Burns School of Medicine at the University of Hawaii, and I took a big sidestep academically. I was studying HIV and human immunology and next-gen sequencing. So that's what I did there. So you're starting to see like the patchwork of random stuff that I've done. I did a couple of years at the Research Corp of Hawaii, and I decided I really wanted to go back to industry. So at that time, the perfect job opened up for me at Pioneer, and I spent the next 5 years leading a team and trying to take pollen precursor cells, microspores, and fool them into becoming embryos, which can be a component of breeding technologies. It's doubled haploid technology. So I did that for a number of years, and then I took an even bigger leap. I decided I want to go get an MBA. I don't know anything about business. I've never taken a business class as a scientist. It's time for something new. So, and by the way, Corteva is filthy with scientists who have MBAs, so really I was just following the path set for me by many other people. And I actually left the lab at that time. So I moved out of doing my own research and into helping scientists at Corteva. So I was a part of the R&D Operations, and I handled a number of roles there. The one that I did before I left is the most relevant here. I did technology acquisition and alliance management. So we called ourselves TAM because we like our acronyms in multinational companies. And TAM was basically business development for scientists. So I worked with scientists in the R&D looking for tech for them, looking for near commercial opportunities for the business in the crop protection side of the business, where it started on the seed. And so there it was biologicals. Biologicals are my jam. Small molecule natural products, regulatory ecotox, and seed applied technologies. So this is the path, the background that I come to you with. And 2 years ago, I made a big jump and I moved into venture capital. So now I am a scientist/investor, but not a finance bro, as I was just mentioning to Pat. Maybe someday. I'm going to tell you a little bit about the venture fund that I work for because it's relevant for this conversation. So I work for a venture fund called Fulcrum Global Capital. And Fulcrum is an early stage AgTech investment fund. What do we invest in? We invest in tech that boosts global food production. We invest in tech that reduces waste in the supply chain. And we invest in tech that does either of those things more sustainably. So that means that we'll invest from the lab bench to the farm gate. We'll invest in the supply chain past the farm gate. And we draw a line at things that touch consumers. We're not food tech investors. So broadly within AgTech, we invest in crop sciences, in animal health, animal production, in robotics, automation, software, water tech. These are the kinds of things that we like at Fulcrum. And now I'm going to share some fun investor metrics with you because you might be curious. So our current fund is $86 million. And from that $86 million, we cut checks between $1 and $4 million to startups. And the startups that we invest in are primarily Series A. So they've done a lot of technical de-risking. They're starting to form some partnerships. Maybe they're starting to pull in customers, but they haven't hit their logarithmic growth to become a big company yet. So that's our sweet spot where we like to invest. I think the coolest fact about Fulcrum Global Capital is, and the reason I joined them, is where their money comes from, where our money comes from. So 70% of our fund comes from farmers. And when I say farmers, I mean they are large elite producers located in a 15-state region in the Midwest. Communally, these farmers represent 30 million acres of row crops, 8 million acres of pasture and grazing, millions of animals under management, cattle, swine, poultry, dairy. And then they also represent auxiliary businesses in agriculture like banking and trucking and ethanol production, capital equipment dealerships, crop protection distributorships. Whatever businesses they need in their ecosystems, they usually build it for themselves. So that's where our money comes from. And with that money, Fulcrum will sometimes call itself the venture arm of the American producer. We're taking farmers' money and we're using that money to drive ag tech forward so that they can see tech in the next 5 to 10 years on their own operations. So that's how Fulcrum invests. So I was asked by Pat to provide some of our greatest hits to give us some legitimacy. What are Fulcrum's greatest hits? One was CoverCress. One of our first investments was in CoverCress. Who's familiar? Okay, almost a lot of you. Okay, so CoverCress, novel GM crop. It's pennycress. It's a cover crop, but you can harvest the seed, and the seed has a really good oil seed profile. The thing I love about CoverCress and its exit is that they had to bring the whole value stream together to get that startup off the ground. It ended up being acquired by Bayer, seed, and jointly, Bunge, crush, and then also Chevron, the eventual buyer of the biofuel. So in order for a new crop to come to life, you need a lot of big, heavy-hitting players to join up. One of our other exits was a company called Telesense, which did grain quality monitoring for shipping and transport. Obviously, not necessarily interesting on the multinational ag side like AgCP, but very interesting downstream from a farm operation. They ended up being acquired by UPL and integrated into part of their business. So those are our 2 exits. We have 12 portfolio companies today. One thing that I'll mention is that AgTech is a unique sector. The exits in AgTech are different than deep tech. They're different than pharma. They're different than software. Why? Because you don't make that much money. How many billion-dollar ag unicorns can you think of that were really successful? Probably less than 10. So the way that our fund invests is that we invest with the assumption that an exit for an ag startup will be between, at best, 200 to 300 million, and that exit is not going to be an initial public offering or an IPO. It's going to be merger or acquisition with one of the large multinational companies. So that's the perspective that we take on investing in AgTech. So with that background, I would like to give you guys a journey into VC 101. How many of you guys have worked with venture capitalists before? Okay, one. Okay, one. Just stop listening. You'll be very bored. So as a scientist who moved into venture capital, I find it an interesting organism to study. And I'm going to share some of my learnings with you in case you're curious or in case you ever need this knowledge. Number one, check out the book Venture Deals by Brad Feld and Jason Mendelsohn. The subtitle of Venture Deals is How to Be Smarter Than Your Venture Capitalist and Your Lawyer. But funnily enough, anyone who moves into VC reads it too, so they also can understand their own industry. Venture is a funnel. You guys know funnels, right? It's like the basis of all science. You got a lot of hits at the top, and then only a couple things maybe that work, hopefully. Venture is a funnel. So at the top of the funnel, you have a lot of startups. You have pre-seed stage startups. You got seed stage startups. You have Series A startups. Then A, B, C, D, and so on until you get to an IPO, maybe, hopefully. We sit kind of near the top of the funnel at Series A. And our fund, there are 6 of us who manage this fund, we will see probably 600 deals per year. When I say deal, translate to a startup. We will get pitches from 600 startups a year. So we get a really broad look at what all of the new tech is that's on the scene in any one snapshot in time. And then how do we vet those 600 companies and make decisions about where we think we'll find return on our investment? The first thing that we do is a series of nesting doll conversations. So the first conversation might be just with me, and then I'll probably bring in one of my colleagues, like, what do you think of this one? Do you like it? And maybe they do, and then all 6 of us will sit down and watch a pitch together. So now we've seen this pitch 3 times. We've tortured this poor startup with 3 different meetings. And this is the point where we have a scoring rubric. Yay, rubrics. Everyone loves rubrics. So do we. And we go through the most important things that we want a startup to have at a Series A stage. What are those things? Those things include, 1, market. How big can it be? Is it really little? Is it really big? I want it to be really big to put my money into it. 2, team. As a scientist who used to do collaboration management, I was so traumatized by this. I can't tell you how many times I've heard my bosses say, I'd rather see a B technology and an A team than an A technology and a B or C team. I found that just personally a little traumatizing because sometimes the science is so beautiful and so elegant, but if the team isn't going to be able to commercialize it, it's not going to make it past the next stage in that big funnel of opportunities. We also look at the technology itself and what the moat around the technology is, what will protect it from other competitors. And we look at commercial traction that they've already had, commercial traction or business development with partners. We like to see those indicators at Series A stage. And we think about the exit. What is the exit? Is it an IPO? Answer, no, it's not. It's M&A with some big multinational company, realistically, probably. And then we also look, this is the part that I always find really, really heartbreaking, at the deal economics. We're a financial investor, so we expect to return 10x of our fund to our investors. And that means that sometimes the deal economics are such that we can't get in the deal. For example, if my exit is at best $300 million, and a startup has already taken in $150 million because it's a super cool robotics company, me investing $2 million into it means I'll own this much, and I won't actually get very much return. So that deal is very exciting, but I can't do it. I have to do something at a different valuation. So something where they've taken in much less money, like 10 million or 15 million. That's probably where we would be investing. So that's the scoring rubric, just for us to decide if we like this startup enough to go forward. The next stage is just entering diligence with the startup. Sometimes we go into their data room and poke around. Sometimes we send them a 5-page Q&A and they send us back 100 pages to review. But we have more conversations with them. We ask them hard questions. We also want to talk to their existing investors. We want to talk to their customers. We want to talk to their partners if they're collaborating with folks. And we take all of that information and the people that we talk to, and then we decide if we still like the startup. And then if we really, really do like it, then maybe we'll offer them a term sheet where we say, hey, we will buy 10% of your company and for this much money, and then we'll sit on your board of directors. And sometimes we join other people's term sheets if a different venture capital fund has offered the term sheet. And at that point, we enter true diligence, where we actually hire people to kick the tires. So then we would hire technical consultants, we would hire legal and financial consultants to make sure that what the startup tells us is accurate. So that's the VC funnel. How many companies do you think we invest in a year after that funnel?
20.
Okay, we got 20. We got 3. Anyone else? It's not 100. It's between 1 and 3. So at Series A stage, we will make an investment in those 600 deals in between 1 and 3 companies, which is why anyone who engages with VC is going to hear a lot of no's because they don't invest in very many. So that's a little bit of background on venture capital. So you understand what you're working with if you ever decide to commercialize technology. But I also thought that maybe the most interesting thing that I could share with you guys would be pro tips on commercializing technology. I know a little bit about this, but I'm not an expert, and I've never been a startup CEO, so I've never done it. But I crowdsourced some information from some of my friends that I thought would be useful to you guys. I'm going to introduce my friends, even though they're not here right now, because I told them I would call them out if they gave me some information. Friend number 1, Vahid Adoun with Corteva Agriscience. Vahid has been in tech transfer on behalf of multinational companies his entire career, 20 plus years. Started at Cargill, went to Syngenta, ended up at Dow Agrisciences, which became Corteva. So he's been representing Corteva alongside startups and academics for a really long time. So he had some really good pro tips and a very funny example. I also consulted another friend from Corteva whose name is Gwendolyn Humphreys. Gwendolyn has a slightly different background than Vahid. She actually spent a number of years at Northwestern University in tech transfer and a number of years before that at the University of Illinois of Urbana-Champaign. And so she brings like a really cool academic background. So between Vahid and Gwendolyn, thank you guys, I have got 6 pro tips for you guys. Okay, I'm gonna list them. I don't have a slide, sorry, you can't take a picture of it. I'm gonna list them for you, and then I'm gonna go through them each one at a time, which I have time to do. Okay, pro tip number 1, know your market. Very important. That's why it's number 1. Pro tip number 2, know your value proposition within that market. Pro tip number 3, communicate. You will have to do it a lot. Pro tip number 4, manage your expectations. This is going to be a fun one. Pro tip number 5, leverage the ecosystem. And pro tip number 6, I just did 6. Okay, 6 for real, persist. So going through these pro tips, the first one, you must know your market. I think you would be surprised how many startups we talk to who don't actually know their market, which it's probably the most important thing for them to know because it affects all of the negotiations they will do, even if they don't know it. You need to know, where will you first commercialize your tech? Will it be geographically limited? Will it only be interested to some businesses? Will it have really broad applications? You'll need to know what kinds of challenges within that market that your tech has to overcome and what has been done before. It's really important to know a market. And I have an example from Vahid that I found ticklingly funny about market size. So with that, there is a university who's got 2 technologies that they want to shop to a multinational ag CP company who shall remain nameless. And 2 technologies. And they're pretty sure that both of them are going to tickle this multinational company. And they're not sure which one is going to hit. But again, they've got 2 technologies. They're basically the same thing. They're both elegant science. One of them I really hope no one from this university is in the room right now. One of them is a transgenic peanut that is resistant to white mold. And then the other technology is a soy yield increase gene with a genetic marker natively in the plant identified. So we got two things. We got peanut and we got soy. This is where whoever commercializes this tech needs to know their market. On the one hand, how many US acres grow peanut? Any guesses? Okay, I see this in the back. Yes, you are correct, but it's still biggish. 1 million. 1 million for peanut. On the other hand, soy, how many US acres?
What?
Definitely more than peanut, accurate. Okay, 90 million. 90 million in the US. So we got soy and peanut. I'm already seeing a difference between our market. I don't know about you guys. Within peanut, only 30% of that acreage is affected by white mold. So now we are down to 330,000 acres. On the other side, how much of soy is affected by yield? 100%, guys. So now we've got 90 million acres that this tech can possibly apply to. But let's also look at a couple of other concepts, like what about acceptance? Who's going to accept our soy native gene that affects yield? Everyone, farmers, consumers, they'll all accept it. What about on the peanut side? That peanut transgene is going to be a GMO, and it might not receive the same consumer acceptance. Maybe it'll be okay in the US market, maybe not so much in Europe. They're just going to have some challenges there. And then lastly, let's talk about who the university is talking to. It's a multinational ag CP company which shall remain nameless, which, by the way, does not have a peanut business. But they do have a soy business, soy seed business. So I think this example illustrates the importance of knowing your market. Obviously, it is an exaggerated example, but understanding the market and where you can actually capture customers is very important as you're selling a technology to partners, to investors, and to customers later down. Okay, so that was number one, know your market. Number two, know your value proposition. This one is a little trickier. From the perspective of many a startup CEO, it is, well, obviously, this has a lot of value. I've been working on this 80 hours a week for like 5 years now. This has a lot of, and it's very elegant science, has a lot of value. Maybe, or maybe not. You know, how is your technology positioned to fill a gap? How? What gap does it fill? And within that gap, are there other solutions already on the market? Are you competing with other commercial opportunities? Even worse, are you competing with duct tape? Right? Because there's going to be some solutions that, you're going to compete with something not intended for it, but it sure is cheap. So that's one part of value proposition. But when you know your other solutions and you canvassed it really well, can you identify your competitors? And can you say to an investor how and why you're better in each of those buckets? That's the part of knowing your value proposition that's really key. I think that at least half of the startups I talk to are not accurate when they show me their competition landscape slide. Sometimes they don't know that they have 3 direct competitors. So don't let that be you. Know your direct competitors and how you're better than them. Okay, pro tip 3, communicate. When you are communicating your startup, let's say you're all going to be startup CEOs with your exciting technology that is very elegant science. You are going to be selling this thing left and right. If you become CEO, half of your time at least is fundraising. You're going to communicate over and over and over and over until you probably sing it in the shower. So it's important to know who is your audience each time you are communicating your message. Is it a technical background group? Is it a financial group like an investor? Is it a commercial group that might distribute your product? Is it a business development group that just wants to collaborate or access your technology? Each one of those groups has different goals and they want to hear a different message. So expect to have different messaging for each one. It's important for you to develop a concise elevator pitch where you can tell someone like me what your technology does in less than 60 seconds. And I have to understand it with no background. So 1, you need an elevator pitch. 2, you're probably going to make a one-pager where you put everything about your venture in one piece of paper to send out to people and hopefully get some interest. And you're also going to have the pitch deck. It is 15 to 20 slides. You might skip some. You're going to go over it many, many times. And it has to stretch between a 10-minute conversation and a 30-minute conversation. So, and it needs to have as few words as possible on each slide, because no one wants to just read the words unless you're sending it in an email and you never intend to present it. And then lastly, within communicate, practice. Practice with your kids. Practice with your financial advisor. Practice with your dentist. Practice with your spouse. Each one of those different people is going to hear different things, have different weird questions, and the more times you answer questions you don't expect, the better you're going to sound when you're asking someone for $10 million, which would be good. Okay, so we got communicate, pro tip 3. We're going to move on to pro tip 4, to manage your expectations. I'm just going to move straight from communicate into bullet point 1 for manage your expectations, and that is one conversation does not equal one commercial deal. No. You are going to have a series of building conversations for anything you want. You want an investor, you will have a series of building conversations. You want a commercial deal, you will have a series of building conversations. You want to out-license someone else's university tech, you're going to have a series of communications. You will never have that first conversation give you what you want. So build your communications such that you don't just give it all right away. Maybe give little pieces, and then you save a little bit for later for those next conversations, because you really just want the next meeting. You want the continuation of the conversation over time. That means you've got engagement. Next bullet point, companies are competitive. Companies are competitive. Shocker, I know. What do I mean by this? I mean that if you are going to a multinational ad company, Corteva, Bayer, Syngenta, BASF, and you want to do a proof of concept collaboration, you might be doing that under non-disclosure terms. And you also might not be allowed to share the data you generate with other folks. Why? Because the multinational that you're working with has put time and money into you, and they don't want you trotting down the street to give it away for free to one of their competitors. Similarly, if you are doing a large-scale joint development agreement as a startup with a multinational company, same thing. You're probably going to have terms in there along the lines of first rights of refusal or exclusivity. Why? Because again, they put time and resources into you, and they don't want you trotting down the street and giving it away to their competitors for free. So don't be surprised when you see those sorts of things, because companies are competitive. Lastly, here in bullet point 4, which is manage your expectations, multinational companies are not service providers. You cannot go to them and say, hey, I'd like you to trial my tech, and I'd like to get data out of that so that I can bulk up my own value proposition and I know what I have. Instead, you must build value and have data of your own to go solicit the interest of those multinational corporations who are not service providers. So always have data that you own that you can share with whoever. Because you can't tell them, oh, I did that project with Syngenta and it was really great. Trust me, they might not. You will need to show them some data that you own. So with that is to manage your expectations. And the next one is to leverage the ecosystem. If you are in academia, you probably have a really great center for entrepreneurship at your university with a lot of people. Some might be entrepreneurs in residence, some might be tech transfer folks, and you can leverage the knowledge that they have and see what resources they might have. Maybe they know about grants that might be really great for the technology you're consider commercializing. Similarly, the tech transfer teams are really great sources of information, not just because they are salesmen for your tech when it becomes IP that the university owns, but also because they know everybody. They know all the other tech transfer teams everywhere. They're really well networked, so they can usually provide introductions that you might like to other organizations. And then similarly, you can also leverage incubators and accelerators. There, you will probably find mentors, you probably find access to investors, you find people who will help you practice and stretch your CEO fledgling wings. But sometimes incubators and accelerators are going to take a piece of the pie. They might take a little bite of equity. So be sure that you understand what you have to give up to get the mentorship from something like an accelerator. And then the last thing that I'm going to share right on time is to persist. Just like being a child actor, if you are commercializing your technology, this is going to be exactly the same. Are you ready? Just like being a child actor, you are going to hear no over and over and over again. It'll destroy your self-esteem, but you will have to dust yourself up and say the next conversation is a different conversation, and you must persist and share people what your value proposition is, even if you feel really bummed out from the no's that you're getting. Because some of those no's are going to be, well, it's not the right time, or you guys aren't at the right stage, or I really want to see you get this regulatory approval. So sometimes those no's turn into yeses over time. And those are my 6 pro tips for commercializing your technology. I had another section, but I think I'm going to pass on it today. But I will say I love startups to death, and it's actually why I moved into venture capital. I love working with startups and seeing their struggles and trying to help them network and overcome those struggles. And if you're considering commercializing your tech, I am in your court. You can probably maybe do it, and you should at least try. You should at least try. So with that, if you guys have questions for me, I will stand up here in the hot seat.
Any questions so far? There's a question to the right.
Thank you so much for that very important discussion. As I said, my startup, I have a startup, but it's working in 4 countries in Africa, and we're doing a POC in New York and in Nairobi. But now, can you tell us about the commercialization from one country to another? Because we find a lot of problems.
Yes.
A need. We find there's a country that wants you to bring their tech in that country, but there's a lot of challenges you face into establishment. So what are the take-out you can give to make sure people can learn from you? Thanks.
Okay, well, I'm gonna start by telling you I'm no expert here, so you should consult like a real expert. Some of the challenges, it depends on who you're working with. Are you working between universities? Are you working between your institution and a multinational corporation? If you're working with one of the MNCs, then they probably have international teams. When I was at Corteva, I was able to leverage regional teams who knew more about the tech transfer than I did. Another one of the big challenges in that space was shipping. You know, if I wanted to trial something in a different geography, just shipping could be really challenging. But again, if you have a multinational partner, they probably have experience to help you with that. I think a university entrepreneurship office has probably seen it before. You just need to find someone who has done the cycle before and can give you some really solid advice for your use case. So your challenge really is to find exactly who has that experience, hopefully in the specific geographies, to answer some of the questions that you have.
Because of the time, we will move along because we still have a moment of discussion with her later on. So thanks, Bonnie, for now. The next talk we are going to have will be from a remote session from Plant-pathogen, and I see Karen is ready for that. So she's going to talk about whole plant physiological identification and quantification of diseases progression. As you notice, my English is not my first language. Enjoy.
Thank you, Vincent. I hope you all hear me well. So we will present to you, and I will present together with Shanae, a new method, as Vincent said, for whole plant physiological identification and quantification of disease progression, which is performed on the plant array system from plant AgTech. We just have some technical difficulty. 1 second, please. Okay. So I'm Karen Moshevion. I am the CEO and partner of Plant AgTech. It's a company from Israel. And Shani, who will present together with me, is a PhD student from the Faculty of Agriculture at the Hebrew University of Jerusalem from Israel. And we will both talk about this subject. I will first introduce Plant AgTech and the plant array technology. And after that, Shani will provide more of the research and application that she is doing during her research about disease progression. So let's start by introducing Plant AgTech. So Plant AgTech introduced the plant array platform, the plant array system. It's a high throughput physiology phenotyping platform. It initially was designed for abiotic stress studies and gained already about 70 scientific articles that were published based on the technology. Some of the customers that are in our customer base are presented here, and we have many more, of course, around the globe. I will describe a bit to you the plant array technology for those of you who don't know us so far right now. So the plant array technology could be envisioned a bit like a smartwatch, only for plants. So what we do, we measure the plant dynamics, so the continuous performance of the plant physiology in response to different environments. And we do that together for many plants in a randomized structure, as you can see in the picture, for the different treatments. And we measure the soil-plant atmosphere continuum. You can see some of the graphs on the right as an example for that. The technology is based on lysimeters or mini scale, very precise mini scales, but we took it several steps ahead, and it's a much more advanced technology than the maybe lysimeters that you know from the old tech. So this is a more detailed description of the system. What you can see is that on the left-hand side in the image is that we are providing personalized treatments, like personalized cocktails for the soil water for each and every plant, could be different. And also we can apply multi-stress, multi-treatments to each and every plant separately and all together, depends on the design of your experiment. In the center of the image, you can see the soil plant atmosphere continuum measurement. All the traits in green describing the physiological measurement, and all the measurements in blue are the environmental sensing, the soil, the atmosphere. We measure the plants about 500 times a day, so it's on a continuous basis. And all of these measured traits and parameters the measurements are being uploaded to a cloud software where it's all being in real time, being analyzed statistically already. So you can see some of the graphs on the right. The idea about the physiological parameters and why we are working with physiological parameters for two reasons mainly. One is that the stomata is very fast to react. So the stomata activity is immediately reacting to any changes in the atmosphere or any stress at all that affect the stomata, of course. And also all these physiological traits that relate to the stomata or also the biomass gain and other traits that we're measuring are directly correlating to yield. So this is helping us a lot, and this is not, we didn't invent this, this is known in the scientific literature, but we are utilizing this in order to shorten the process for screening. So it's fast screening based on the yield potential between the plants. The measurement that we are taking in a comparative manner simultaneously to all the plants. Very quickly, we can identify those that have more drought tolerant or any other traits or any other ability that we are looking for as a researcher. And we are shortening the whole cycle dramatically. We don't need to wait for until yield. And of course, also the depth of the knowledge is increasing dramatically. So it's not the guesswork anymore. It's much more precise and much more informative and quicker. This is just an example of, let's say, a normal graph from our system, from the Spark Analytics software, where you can see the soil water content that we are controlling. So this is controlled by the system. It's actually, you can see that the drought reduction is in the same rhythm, same rate. And this is thanks to the feedback irrigation that we have in our system, a quite unique capability. In terms of how we control the soil water content, we can control it by the soil sensor, we can control it by on-plant transpiration and other ways. Also, you can see then, the different lines, you can see how the different lines behave, the different varieties of tomatoes in this case, very clearly with statistical analysis already. This is another resolution looking on a day variations. Here you can see the continuous measurement over a day. In this case, it's transpiration and stomatal conductance versus the environment, VPD and PAR. And every window of difference, let's say, if you can see a peak here, could end in accumulated effect of about 15-20% for just a peak that looks like for 1 hour or 2 hours. So this accumulates very significant yield impact. The system can work with large variety of plants, from Arabidopsis through vegetable crops and field crops, and also even trees, as you can see here. And there are different applications that are useful to do on the system. It's genetic screening and biotic stress. You can see drought. Also, we can control flood, salinity, and so forth. Physiological studies of different kinds, nutrients and biotic screening, seed treatments and how they affect the whole plant, optimization of agronomical treatments, and biotic stresses. And with this, because we're going to focus the rest of the talk about biotic stress, I'm going to transfer the discussion to Shani. So I will stop sharing and Shani will share from her computer. Forgive me for a second. Shani, it's...
Okay. Hi. I will present to you a section of my research using the plant array to identify and quantify disease progression. My name is Shani. I'm a PhD student at the Hebrew University in the Department of Plant Science. I've always had a deep love for plants, but I have to admit I've unintentionally become quite good at killing them in all sorts of ways. But funny enough, the lack of causing stress to plants actually is the focus of my PhD research. Let me introduce you to one of the top 10 fungal pathogens, Fusarium oxysporum. This forma specialis is a soil-borne fungus that specializes in attacking tomato variants. It causes a disease we call Fusarium wilt. The infection starts at the roots and moves up into the xylem, where it blocks the water flow. This leads to wilting, yellowing of leaves, and death. Fusarium oxysporum can devastate tomato yields. It can impact both commercial farms and home gardens. All of our Fusarium work was done in collaboration with Dr. Shai Koba from the Department of Phytopathology. To inoculate our plants with Fusarium, we use a common protocol. 3 weeks old seedlings were cut at the roots and submerged in the Carnegie suspension of million kinesiads 2 ml. Then they were planted in 4-liter pots on the plant array system. How do we assess the Fusarium wilt disease? For the first few days, you can't see anything in the inoculated plants, like this picture. This picture was taken 11 days after the inoculation, where you can see this leaf is missing, this leaf is yellowing, and some leaves are not fully open. As a scientist, I need to quantify this tiredness and yellowing. How do we do that? Looking at the literature, there are 2 main ways to assess diseases. Usually, an experimenter visually inspects the plants and gives them a score. 0 means no symptoms, and 1, 2, 3 indicate various degrees of yellowing. However, this method can be inaccurate since it's subjective, and I've seen some works talking about the problematic of visual subjective scoring. The other main method is measuring yield or biomass at the end of the experiment. However, I feel this misses some of the disease dynamics because it's only at the end, like 1 point. We want to offer a third option, using the plant array system to assess the disease. By definition, disease is an impairment of the normal state of a plant that interrupts or modifies its vital functions. We have a tool that can continuously monitor vital function of a plant. This includes biomass of the plant, transpiration, the water loss, nutrient use efficiency, stomatoconductance, root activity. It's easy to use. It's quantitative and objective. It's comparative. We can have control and infected plants simultaneously on the system, and it's continuous. From day 1, we can follow the disease. Our goal is to use the continuous measurements to detect the disease in plants early, even before visual symptoms. Additionally, we aim to use the system to quantitatively assess the severity of the disease. Before I dive into the experiments we did and which Fusarium strain we used, or tomato cultivar, I want to present to you this overview. This is a summary of the experiments we did. Every row represents an independent experiment with a specific strain of Fusarium and tomato cultivar. In all the experiments, we followed with the plant array the transpiration and the plant weight, and we came to inspect the plants visually to see if we see any morphological symptoms. In the table is when t-tests showed that the inoculated group is different from the control, same cultivar but with water. In all the experiments, you can see that transpiration was the first to be different between the groups. Next was the weight, and last we saw visual symptoms, if we saw at all visual symptoms. Fusarium grows in the vascular system and blocks it, so it makes perfect sense that the first symptoms we see are in the transpiration. Just from this overview, you can understand that the plant array system gives us an ability to early detect the disease. To cope with Fusarium in the field, farmers and growers use non-susceptible cultivar. Let's talk about plant susceptibilities. There are 4 main words to discuss it. You can say immune, resistant, tolerant, and susceptible. The problem of some of these words are not black and white. How tolerant is a plant? How susceptible is it? We suggest talking in precise percentage of yield, and I'll give an example. This is an industrial cultivar named M82 that is tolerant to Fusarium, and this is Rehoboth 13, which is susceptible to the pathogen. We followed the plant weight after the inoculation, and we could see differences between the cultivars. You can see the trend starting very early on, but we would look at this stage. At gray, M82 control treated plants get to around 220 grams. When inoculated, it loses about 15%. Interestingly, Rehoboth 13 in black, the susceptible cultivar, produces higher biomass, 23% more than M82. In brackets, I would say this is not significantly different. But when inoculated, R13 plants decrease biomass in 60%. There is a high correlation between biomass and yield. And now that we have this precise percentiles information, A farmer could decide which cultivar he wants to use. If he wants to take a risk and grow R13 and perhaps gain 23%, or he knows he has Fusarium in his field and he is satisfied in losing only 15% with M82. This tool is quantitative, exact numbers, and it's early on. You can see the trend starting early on. We did this with 2 other cultivars, Marmande Verte, I hope I'm saying it okay, a susceptible cultivar, and Mutella, a resistant cultivar. We followed the transpiration of these plants, and here too, the resistant cultivar in gray is acting nicely. When infected, it loses some transpiration. The susceptible cultivar in black doing better when uninfected, but losing much more when inoculated. I can present this graph one other way, taking the daily maximum for each day and for each plant, calculating the relative transpiration. This graph, relative transpiration from the daily maximum. I found this presentation very interesting because you can see nicely how the percentiles stabilize and perhaps how the system can help early on quantify the differences between the cultivars. I talked about differences in tomato cultivars. Next, I want to talk about differences in fungi strains. One major question in phytopathology is quantifying fungal virulence. This is an experiment using 2 Fusarium strains, a commonly used lab strain, we call it moderately virulent Fusarium, MVF, and one we got from a seed company that collected it from a contaminated field. We called it virulent Fusarium. We did a traditional seedling assay proving the differences between the strains. In traditional methods, you take tiny seedlings and inoculate them. Then 3 weeks later, you score the symptoms. This is picture 3 weeks later. And you can see that the virulent Fusarium inoculated plants were dead or wilted. The inoculated plants with MVF, with the other strain, showed also wilting, some disordered, and some look healthy but do have browning in the vessels that indicate the Fusarium growth inside. This test worked well, and this is a graph representing the different groups. But from my experience, it is hard to decide sometimes what group a plant is in and to find the browning in the tiny vessels. Anyway, I want to have something more field-like environment with big pots, with fertilizer, with sunlight. So I went to the plant array system. Using the plant array system, you can see that in greenhouse conditions, after 18 days, the plants still look fine. Looking at the transpiration, at day 18, we could find differences between the control group and the virulent Fusarium. After 22 days, we found differences between all 3 treatments. This is showing us nicely the disease progression. And the best thing about it is that it is before visual symptoms. This is a disease score we gave visually, and it only occurred after day 22. In the last part of my talk, I will shortly introduce you to 3 other plant-pathogen interactions that were monitored using the plant array system. The first is Phytophthora infestans that causes the late blade in potatoes. In the field, the first symptoms are usually these spots on the leaf tissue. We inoculate the plants by spraying them with a pathogen suspension and at the date where the arrow is showing. Using the plant array system, we followed E. E is the transpiration normalized to the plant weight. So 3 days after the inoculation, you can see here was the inoculation, 1 to 3 days after inoculation, there is a significant difference in E between the control in gray and the inoculated plants in black. In the picture that Mendel took, you can see the disease is not yet showing in day 3, only later. Another disease that starts in the leaves is the tomato yellow leaf curl virus. This work is of Mishra's, and he took healthy and well-watered plants, and they transpire nicely up to 800 grams. But already from day 1, virus-infected plants transpire less. Interestingly, they tested how virus-infected impacts drought tolerance. When exposed to drought, virus-infected tomatoes are more resilient than uninfected plants. The inoculated plants reach the critical point later. You see the healthy ones the healthy ones got to the route earlier than the virus infected. So in the pictures, you can see the healthy plants, how they look really bad, and virus infected are still standing strong. The last use case I will present is Wexler's work. He had an experiment with peppers that had some kind of root rot. By the end of the experiment, the shoots looked quite similar. However, the roots showed varying degrees of rot. So they classified the plants into groups of root severity. When using this grouping and looking back at the daily transpiration, they could have noticed differences between the plants that were found to be infected and the healthy plants. The difference was found already here, much before the end of the experiment. Interestingly, they compared these findings with reflectance spectrum measurements across different stages of root rot. I know many of you are in the imaging area with different drones and spectrums. I believe the first step is to fully understand how infection disrupts whole plant physiology through these continuous parameters. And by placing imaging systems over the plant array, we can identify physiological indices, how physiological indices impact thermal reflectance and measurements. I find this can be an important step towards better understanding field sensors and drones. To conclude, the Lizimeter system enables early detection of disease, even before visible symptoms appear. Transpiration is the most sensitive physiological trait we can monitor, and this continuous system is ideal for quantitative comparison between plants and fungi and may be utilized for breeding plant disease resistance. These days, our work is in peer review, and we would like to hear your questions and feedback. But first, I want to thank my lab members and workers and my family and friends for supporting and helping me in the greenhouse. And I'm sorry I couldn't attend the conference, but I'm grateful for the opportunity to show my work. And I want to thank you for making this possible.
Well, thank you very much for this presentation. Are there any questions from the audience? There are none? Do we have none questions, ladies and gentlemen? There is one.
Thank you very much for the talk. It was an interesting talk. I was curious if you are able to use this for the disease testing. How do you deal with the cleaning after this experiment? Because it's, this machinery. Can you explain a little bit about that?
Yes. All the pots and the sand goes to with chlor to autoclave, and all the pots go into big mechalim. Big tanks with chlor, and everything is really needs a lot of work to in the end of the experiment to be clean again.
Any other questions? No questions? That's also okay, but then I thank you very much for your presentation, and then we're going to the next one. Thank you.
Thank you all. Thank you very much.
Okay, ladies and gentlemen, we are going to take off because we will have Samuel here and telling something about drone sampling for phenotyping applications.
Okay, it works great. Hello everyone, I'm Samuel Oswald. So I'm working at Vito. We're a Belgian research institute, and I'm in the remote sensing team. We're a large team which works on all kinds of different applications involving drone and satellite imagery. As the title of my presentation suggests, I work on drone applications for phenotyping applications. So I'll get into it. I wanted to start by showing a few images. Here's a very high resolution RGB image. You can see like a lot of details of the plants taken very close up. You could spot spots on the plants or other kind of features if there were any. For another one, here's a multispectral imagery. Multispectral in drones, if you've ever worked with them, tend to be at a lower resolution. typically 1 to 2 centimeters or lower, whereas this is also at a very high resolution, maybe what you would expect with RGB imagery. So how do we take these photos? You could probably think of a few ways. You have custom bespoke machinery, like in the first image, which can fit all kinds of imaging devices and other things. You might use a very high-end drone, which can support a high payload and take high-resolution images from a height. Or you could, of course, send PhD students into the field on bicycles. The trouble with these methods is, in most cases, they're not scalable. They're all either quite expensive or you're limited in the amount of PhD students you can exploit. And so what we have been working on at Vito is a new method based on our own kind of interaction with researchers in the phenotyping domain, and we call it drone sampling. And the idea is to get this super high resolution imagery at a low cost. So likely a lot of you are familiar with this when you're doing a field drone campaign. You typically want to get a high amount of overlap and you work across a whole field. The idea being that you use structure from motion to recreate your image. And there is a lot of different outputs you can get, plant cover, plant height, plant health with multispectral. And you can also start using your object detection methods to start counting plants and this kind of thing. And so this has been fine for a while, but as you move into high resolution, you come across two key problems. The first one is that it requires a high image overlap. As I mentioned, the structure from motion method requires that you don't just take a photo of part of the field once, you take it multiple times so that you can get overlap of the images. The downside of this is that you then end up with the same parts of your field photographed over and over and over again. And this maybe was fine when we had lower resolution images and it was only a few gigabytes extra. But now if you're working on fields where you might be working at 1 millimeter resolution, this can get into hundreds of gigabytes of extra data that you don't really need and won't use. The second issue, kind of related to this, is of course the anchor points, which is why we need to have high overlap. So when we use structure from motion, we have an algorithm that looks for common matches between images and then use that to reconstruct a 3D model. Typically, for drone missions, we want to look at matching the images between dates, so we'll also align them to various GCP points. If you try to use a camera that's not a very high-end camera and want to use this method to get very high resolution, you'll probably run into a few issues. So on the right with image C, you see how we went trying to use structure from motion to recreate a field using 1 millimeter resolution in red, which just kind of failed because it couldn't find anchor points between all of the images. In yellow at 2 millimeters, which also performed a bit better, but didn't quite work. And then at 5 millimeters, it does work. But if you look at the images on the bottom left, you can see for our 1 millimeter image in BI, we did have high resolution and we could probably identify things like the wheat heads in this case. Whereas for the 5 millimeter resolution, there's not really much you can get out of it. So whilst you cover the whole field, you don't get the information you want. And so that's the kind of problem that we have. So right now, to map a 1 hectare field at 1 millimeter resolution, you'll probably need a high-end drone, which can be 70K, have a 55-minute flight time, and you will end up with about 335 gigabytes of images, which can just be hard to work with. It'll increase processing time. You also have to think about data storage. And if you want to scale this to multiple fields, it's going to become quite difficult. So our proposition then is the aforementioned drone sampling. And so this is made possible through things like the increasing GPS accuracy of drones. So typically in a phenotyping use case, you still do want to really accurately capture your plots. So we suggested RTK drone for this. And if you look in the top, you are probably just interested in your plots and not the whole field. So all of these areas in green, they're probably not interesting for your eventual downstream analysis. With drone sampling, we aim to just take photos of the plots, which will then cut down on all of the extra data, allow you to fly with drones that are more easily available, which are cheaper. You can fly more, you can process much faster. So it can be a big benefit when you do want to get these very high resolution applications. So yeah, how it works, it's similar to any drone flight that you've done. You'll have a planning stage, a flight stage, and a processing stage. In the planning stage, we want to create our digital plot design. And so this is maybe one of the things that you kind of want to work out originally. There's a few ways to do it. You might have a planting machine that allows you to very accurately locate the GPS coordinates of your plots. Or you can do a traditional drone flight earlier in the season, which can be at a lower and therefore easier to achieve resolution, and delineate your field using that. But from there, you will define your plot centroids, and then depending on what you want, you can think about the camera type required, whether it's RGB or multispectral, your required resolution. And then through the workflow we've developed, you can create a mission file, which allows you to upload it to a drone and then fly it. So yeah, this is the kind of comparison with the data requirements that we end up with. If we use this method, you can see that for a traditional drone mission, we're looking at hundreds of images, whereas it just gets down to tens or a few dozen for drone sampling to cover the same area. So yeah, this is just I'm not sure if it's playing, but this is a video of a drone flying with this method. I think we actually end up just having a still of that video, but off in the distance you see a drone. So if you want to see the whole video, it's very exciting, and you can come and find me later for it. Oh no, it does work. I just had to press the play button. There we go. Yeah, so this is just kind of how it works. You can see it's similar to any drone flight, but we don't have to fly it so high, so we don't end up with this need for anchor points, which are typically found between all of your different plots. You just focus on the plots themselves. And I think this one's also animated. Yeah, so from here, you don't need this image stitching. The kind of one thing that you lack here is this kind of plant height, which is obviously a useful thing to get for. That's still possible. It is possible to do a lower resolution and a higher resolution flight, and that can end up being a more affordable option than just doing a high resolution if that's what you're after. But it might be that you're not so interested in that, and you're interested instead in the actual output products themselves. So yeah, you can see that we still now have, with a much cheaper drone, these very high resolution images. And with that, we can look at our extra downstream products like object detection and what have you. So yeah, that was the kind of planning and flight and the processing in the end. I think from here, I can talk a little bit about what some of the things you might be interested in are. So this doesn't just pertain to drone sampling, also normal drone flights, but a lot of them are much more achievable with this drone sampling technique. Here is an image showing your object detection. So that's obviously becoming very popular now with the advent of AI and machine learning in the drone phenotyping domain. Other things might be looking at your kind of plant or gap count. So in this first image, we try to count all of the plants in a plot. but also where you have gaps, which is useful for finding your emergence in the field. As you go to the other end of the growth cycle, you'll probably be interested in what your yield looks like. So this is a pumpkin case where we calculated the size, so the width and length of pumpkins, which is obviously very indicative of what you'll be able to market in the end. Other things might be disease detection, and this is something that you really often do need very high resolution for. I think this is also a video which isn't playing, but now it should be playing. Yes, it's playing. So this is kind of a field that we had before. Initially, we did a lower resolution flight, and then we did a drone flight over it, and it kind of shows you for these plots the accuracy of this drone sampling method. So each of the plots, it is georeferenced over where we would expect it, and so this becomes comparable between dates. Compared to the last image, you can see that we have this very high definition image of the field, and so this becomes usable for things like the disease detection. So in this case, the plots are lined up between the dates, and you can see in some plots the deterioration of the plant leaves and where they remain healthy over time in others. And I think it goes on to show more of this. And of course, there's a lot of other things that we can do. Here's some that look at fruit segmentation. So when you have high enough resolution, you can also look at trying to segment your plant objects. We've discussed a lot about this. I think in recent years, we're moving away from things like just detecting how many objects you have and more towards what do the objects look like, how do they differ, and all of that kind of stuff. Weed identification is also important. I know this has been a topic kind of yesterday in the MLCAS, and also for many of you, since that can be problematic on the fields. And of course, the multispectral use case is also something that's quite interesting. So as we talked about in the past, when you looked at multispectral imagery, it was typically at a lower resolution, so you might be able to get ideas of plant health with your vegetation indices for a whole plant, but because of the mixture of pixels, you couldn't look at a leaf level or try and identify features of leaves. But this is also something that's possible here. So you can see here we have our NDVI for the leaves themselves, and you can see which ones have a high kind of healthiness index according to NDVI and where it is less than ideal. And this can then be related to biochemical processes or diseases. So yeah, I just wanted to then finish off by mentioning a bit Mapio. So Mapio is the kind of end-to-end workflow for drone image processing that we have at Vito. And so the idea is that we're experts on remote sensing, so on things like geospatial image processing and all of this kind of thing. Often, if you are working in plant sciences, this is something that you might work on yourself, but maybe it's not your core area of expertise, and maybe you want to partner up with another institution to do it for you. If so, that can be us. So yeah, that's we take you from the plan through to the flight analysis, connecting to your own downstream applications, and so on. This is kind of just a look within our web platform of what you can see with a traditional drone survey versus drone sampling. So yeah, you can see for this wheat field, you do get much larger coverage using a traditional survey, but usually this isn't really something that you're so interested in. Likely you'll want to get statistics on the amount of wheat ears or the size of fruits or whatever, but just representative of a plot. And so in that case, the drone sampling case will of course be more useful. You can see in the bottom left, a very high-end drone was used to fly this traditional drone survey, Whereas for the drone sampling case, it's a drone that's much more accessible. You can then extract plot statistics, as I mentioned. I won't get into this kind of stuff, but we do have a booth here that we're sharing next to the entrance door with ISTIC, the guys with the cool glasses. So we're happy to take you through a demo of any of the functionalities of our platform. And likewise for the actual planning and how you create these kind of mission files. So to conclude, as we have developed drone technology, image processing kind of capacity, storage and all of that kind of thing, the importance of image resolution for drone-based phenotyping has kind of increased quite a lot. But for a traditional drone survey to actually get this very high resolution, you'll typically need to invest quite a lot, and you'll end up with a lot of redundant data as well as that. And so our methodology is the drone sampling. So yeah, that can really take down your investment cost, the time that someone is required to be in the field, because when you are doing a drone flight, you need someone there to actually operate the drone. And the processing cost is down by quite significantly as well. So you will have results available for further analysis quicker. So yeah, that's my talk. I'm happy to discuss further at the Veto booth. You can contact me via email, and I will also answer questions now. So that's great.
Any questions?
Thank you, very interesting talk. I have two questions related with technical details. First one was, how did you decide the optimal number of sampling rate, or how you downsample the original data?
For the how did I downsample on the original one? In that original image for the downsampling, this was actually 3 different flights, if I understand your question correctly. So we used the same camera. I think that was a Mavic 2 at that stage, or maybe a Phantom 4, I'm not sure, but at 3 different resolutions. Yep, I hope that's was your question.
Yeah, thank you. That's good. And one suggestion for you was to maybe check on an accurate sampling theory coming from signal processing. That has a minimum number of sampling you need meet to guarantee you can reconstruct the signals in a perfect way. The second question I want to ask was, how do you decide your downsampling of the drone data will not affecting your 3D reconstruction or structure from motion, the future 3D model quality?
Sorry, how did my downsampling not affect the structure from motion? Or
yes,
so I think that in this case, I would say it didn't affect it. I mean, also because it was the different flights. For the upsampled flights, I think this is just when you have the 1 millimeter, it's just going to always be a risk. Sometimes you will have a flight at this resolution that does work, but it doesn't work consistently, we found.
Okay, interesting. Thank you.
Thank you.
Thank you so much.
Thank you.
My question is on data privacy and security concerns. How do you deal with that?
The data privacy and security?
Because there's a lot of data which come in, and how do you deal with that data privacy and security concerns?
I mean, all of the data is stored securely. I think that the data ownership kind of depends on the kind of client that we're working with or the partnership that we have. For example, some really are going to want to protect their own data and then it's not shared, whereas others will want to kind of enter a research partnership where they do publish things. So we are kind of versatile, as we're a research institute that's kind of in the applied research domain. We work with both academic institutions, but also private enterprise. And so there's different requirements depending on the job that we're doing. Sorry,
doing data share agreement with your partners, I think is one of the most, and farmers, because that's one of the thing also I've been doing, sharing, having a data share agreement with farmers that they can be comfortable with their data, make sure you can be able to use that.
Indeed.
Diagnostics and stuff. I think that's one of the thing I use also.
Yeah, indeed. I think, the data sharing with farmers is important. For this, it's not often strictly our kind of domain to share the data with farmers, as we do work with other researchers and with plant breeding companies, other researchers. So often this is something that we work out with them.
Let's have one question.
Thank you for your presentation. My question is, did you work about real-time data analysis by drone? I mean, did you put some custom software in the drone? After then, the drone gets an image. After that, the same time, data analysis.
No, so we don't do onboard real-time processing for the drone imagery. I think I understand what you're getting at, where you could kind of, try and isolate the plot within an image and then take the image at that point. I think that's a future step that's very interesting, but not what we're looking at now. So how it works now is that we look at the kind of the plot centroids, and we already have a predefined flight path that we utilize.
Okay, thank you so much.
Thank you. Okay, thanks a lot.
Thank you very much for this talk. Now we're going to continue with our well-deserved coffee break, and then at 16:25 military time, we will continue. See you back.